This unit is about MHC, and Rittenhouse actually covers MHC in 2 different chapters. So we're going to look at both chapters in this unit. We may run into some redundancy, but that's okay. We'll deal with it. The MHC complex is an area of the genome that encodes for the HLA antigens. It was discovered in the early days when they started to transplant tissues and organs from one person to another. Of course, they ran into a rejection problem. Even after they figured out that about the ABO typing and checking for ABO compatibility, they still had rejection issues with the transplantation of foreign tissue. So they looked closer and discovered the presence of the MHC. The MHC molecules play a role in both the humoral and the cell-mediated immunity. Only class 1 and class 2 MHC genes play a direct role in the immune response. There is another class 3, that encodes for cytokines and complement and heat shock proteins. The human MHC antigens are involved in the immune response. They're also called the human leukocyte antigens because they are found on white cells. They encode for proteins on the antigen presenting cell, and of course that's the cell which presents the antigen to an appropriate T cell. We can nitpick and we can stipulate that MHC means the particular location on the genome, and HLA are the products that are coded for by that genome, but most people kind of just use the two terms interchangeably, and that's what we'll do in class. The MHC haplotypes are highly polymorphic. They have many different alleles, and these alleles are inherited as sets. Each offspring gets one set of alleles from mom and one set of MHC alleles from dad, and then these are codominantly expressed. They all express their products in the offspring cells. Here you see what's going on graphically with these MHC genes. So from mom up here, mom's got 2 chromosomes, so she's got 2 sets of MHC alleles represented with pink and orange. So each one of her offsprings is going to get either the pink set or the orange set on their cells. And then there's dad with our blue and our green set of alleles. And so each offspring is also going to get one set from dad, so they end up with either the blue set or the green set. And then all of these are expressed on the offspring's cells. The MHC class 1 molecules are involved in antigen presentation of endogenous antigens. These are ones that end up inside the infected cell, such as viruses. They're found in all nucleated cells, so of course that excludes the red cells. They're encoded by A, B, and C regions of the MHC, and once they present the antigen, they are presenting that antigen to cytotoxic T cells, which are going to then be involved in the killing of the infected cell. The MHC class 2 molecules are found on antigen presenting cells. They're encoded by the DR, DP, and DQ regions of the genome. These recognize exogenous antigens. These are extracellular antigens that have been engulfed by an antigen presenting cell. They present the antigen to helper T cells. Looking at the 2 different types of MHC molecules, over here on the left we have an MHC class 1 molecule consisting of 3 alpha domains, alpha 1, alpha 2, alpha 3, and then there's a beta 2 microglobulin area nestled off to the side with 1 point of attachment to the cell that possesses it. Note that up here in this area is the antigen binding region. Over on the right, we see an MHC class 2 molecule with 2 alpha regions, alpha 1, alpha 2, and then 2 beta regions, beta 1, beta 2, and 2 points of attachment to the cell. But again, out here on the end, and this is vaguely reminiscent, I think, of the structure of an antibody with the arms sticking out. Out here at the end of the arms would be where the antigen would bind. On the surface of T cells, there are certain receptors, which we call T cell receptors. The T cell receptors also share some structural features with immunoglobulins in that they're composed of 2 heterodimers covalently linked by a single disulfide bond. They have a variable region for binding antigens, a single immunoglobulin-like constant region, a membrane-spanning region, and a hinge-like region. These receptors are associated with the CD3 marker, which signals and activates the cell after antigen bind to it. The differences between T cell receptors and immunoglobulins: the T cell receptors exist only on cell surface. They're not ever secreted like immunoglobulins are. Somatic mutations do not contribute to the repertoire of the various T cell receptors, and there's no rearrangement of the constant region genes like there are with the immunoglobulins. Antigens that are taken up by antigen-presenting cells tend to be strong immunogens. In other words, they're very efficient at stimulating the immune response. They cause the activation of CD4 and CD8 T cells, and this is compared to antigens which are in solution, which are typically weakly antigenic. So what happens is that the accessory cell or the antigen-presenting cell binds to a particular antigen. It takes it inside. It internalizes it. Once it's inside, it breaks it up into various parts and pieces. Then it takes one or more of those parts or pieces and takes them to the cell surface and attaches them to either its class 1 or class 2 molecule, depending on the type of cell that it is. It's kind of like it takes a prisoner, chews the prisoner up a little bit, and then takes a portion, let's say it takes the prisoner's left hand, okay, kind of disgusting, but takes his left hand and he holds the left hand out on the outside of the surface and he displays it for all to see. The interaction of the peptides with the MHC class 1 and class 2 occurs at different sites in the cell. The class 1 and class 2 are transported to the Golgi compartment after they're synthesized. Class 1 and class 2 segregate, where class 1 interacts with antigen peptides and class 2 are complex with the invariant chain of the molecule. The class 1 and class 2 molecules do different things with antigens. The class 1 molecules bind the antigenic peptides in the endoplasmic reticulum. These are processed fragments of proteins from something infecting the cells. However, the class 2 molecules travel from the ER to an endosome or a lysosome, and there they interact with the antigenic peptides. Typically, their phagocytized antigen is brought in by ABC proteins and transporters associated with antigen processing molecules. And these are the TAP1 and TAP2 molecules, and then the antigen presentation follows. We'll look at a case study that has to do with the TAP transporters being abnormal. The quantity of MHC class 2 on monocytes and macrophages varies. Interferon gamma released by activated T cells causes an increase of class 2 molecules. This is a nice chart summarizing the nuts and bolts of the MHC class 1 and class 2. So take note of the fact that the MHC class 1 molecules interact with CD8 positive T cells. These are the cytotoxic T cells. The type of antigen involved with the class 1 molecules are the endogenous antigens, some bacteria as well as viruses. They're found on all nucleated cells of the body. Antigen size tends to be a little smaller. Peptides, 8 to 10 amino acids. The antigen is added to the MHC molecule in the Golgi apparatus. Looking at class 2 molecules, they react with CD4 positive T cells, which are the helper T cells. These interact with exogenous antigens. This is most bacteria that have been phagocytized by phagocytic cells. The found on antigen presenting cells, such as cells of the monocyte and the macrophage lineage, as well as B cells and dendritic cells. Peptides a little bit longer, 13 to 18 amino acids. The antigen is added to the MHC in the endosomal compartment after the invariant chain has been removed. Now we're going to look at the portion of Chapter 14 that deals with the MHC complex. So in Chapter 14, the major histocompatibility complex antigens, we refer to them as MHC or HLA. These are the major components seen as foreign by the recipient of a graft, tissue, or organ. These genes are the most polymorphic genes in the human genome. Each locus or location on the chromosome has a very, very large number of different alleles. Matching donor and recipient is a challenge unless the donor happens to be in the same family as the recipient of the organ or tissue. The HLA system is inherited following a typical Mendelian inheritance pattern. Each offspring inherits one haplotype from each parent. These haplotypes are inherited as a set of genes that are usually transmitted together as a single entity. As children have no more than 50% compatibility with each parent unless the parents happen to be very genetically similar. Two offspring of the same parents have approximately a 25% chance of inheriting both the same haplotypes. Here again we see our three groups of MHC class antigens. MHC class 1, the A, B, and C. MHC class 2, the DP, DQ, and DR. Both of these are involved in transplantation. And then the MHC class 3 grouping is involved in other activities. This is similar to the diagram we looked in the other chapter, where each offspring gets one haplotype from each parent. What they've done additionally is draw in a situation that occurs, albeit rarely, where there is crossing over. In this particular child, what has happened is these two chromosomes have crossed over, and so this child has received half and half of mom's haplotypes, but that's a rare occurrence. In the laboratory, histocompatibility testing and matching is critical in almost all cases of transplantation in order to minimize the rejection of the transplanted tissue. So what we need to do is address the histocompatibility profiles of both donor and recipient, and there's several steps involved. Step 1 is test for ABO compatibility. If the organ or tissue is not ABO compatibility, there's slim to no chance that the transplantation will be successful. And once we establish ABO compatibility, then we look at HLA compatibility. We can consider this as kind of a three-step process. First thing we want to do is type both donor and recipient for their HLA antigens to determine how closely they are matched. Then we'll need to test the recipient to see if they have any preformed antibodies against any antigens that are present on the donor. How could these preformed antibodies have been stimulated? Well, if the person had had a previous blood transfusion or a previous transplant, they could have been stimulated to produce antibodies. And then lastly, we do something very much akin to a blood bank cross match, where we take individuals and mix cells together to see how well they play together. One type of HLA typing that was done historically, not used too much anymore though, but this test is called the microcytotoxicity test. For those of you who are experienced blood bankers, this is somewhat akin to typing red cells for the various red cell antigens, except of course we're testing white blood cells. So we take the white blood cells and incubate them with various antibodies to class 1 and class 2 antigens. After an incubation period, we add complement. The cells that possess the antigen to the antibody that's been added are going to be killed by the antibody and complement. Then we add eosin, which is a viability dye. Eosin is pink, and the only cells that will take up the pink eosin will be the dead ones. So after staining, we take a look at them, and the pink cells are the dead ones, indicating a positive result in that those cells had the matching HLA antigen to whatever antibody was used in that particular test tube. Couple of drawbacks. This is naturally limited by the number of commercially available antibodies. So we know there's lots and lots of these HLA antigens. There's not that many HLA antibodies available to match them. And the other drawback is that after death, the HLA expression diminishes, so that the test is only valid on living cells. And of course, a lot of our tissues and organs come from cadavers that we intend to use for transplant. And so this test method wouldn't work at all for cadaver tissue. This graphic is showing us what goes on when we do this microcytotoxicity assay, whereby we take cells that are going to be tested for their HLA antigens, and in each different test tube containing these cells, we add one particular HLA antisera, the antibodies to a given antigen. We allow them to react, and then we add complement. If the corresponding antigen is found on the cells, then the antibody would have complexed with it, and then when we add complement, the cell will be killed. So after that is allowed to transpire, we stain the cells, and only the dead cells will take up the pink stain, so that pink cells indicate a positive result, meaning the tested cells contained the corresponding antigen to whatever antibody was put into the test tube. A negative result, no pink cells, meaning that the tested cells did not possess the tested for antigen. Back in the day when the microcytotoxicity assay was in use, it wasn't good enough for certain types of transplants, and notably in bone marrow transplantation, the limited resolution, in other words, the limited number of antigens we could test for, was problematic. So in those days, we added another test to the test battery before considering transplanting that tissue, and the other test is called the mixed lymphocyte reaction. In the mixed lymphocyte reaction, which is historically relevant, not used very much anymore, in this test methodology, lymphocytes from the donor and the recipient are mixed together. If those lymphocytes are not compatible, they'll stimulate each other to proliferate. We assess the amount of proliferation by using tritiated thymidine. The thymidine would be taken up by the proliferating cells in the making of new DNA. So we could monitor the thymidine uptake, and the more thymidine that's being used, that relates directly to the amount of new DNA that's being produced, which then correlates with the amount of proliferation. A definite drawback of this test methodology is it requires several days of incubation. On the other hand, it does detect cell activation. What it's doing is not identifying HLA antigens on either donor or recipient. It's rather just throwing them together and see if they get along. And if they get along, that indicates that they are relatively closely matched. If they don't get along and they start proliferating, they're not a particularly good match, and therefore the transplantation is probably not going to be a go. A drawback on this one as well is that it doesn't work with cadaver donors, and because of these drawbacks, it's been largely replaced by actual HLA genotyping of recipient and donor. In today's world, we tend to test for the actual genetic makeup of the genes that encode for the HLA antigens rather than the products or the antigens. This is done with polymerase chain reaction testing, where the PCR amplifies an area of the DNA and determines whether the sequence of a particular HLA antigen is present or not. It's often carried out, or it is carried out, using sequence-specific primers, SSPs, that detect genes at a particular HLA locus. We can measure specific gene variants in an individual by using specific DNA probes to recognize specific DNA sequences that reflect a particular gene variation. This PCR-based genotyping is much more precise than the formerly used microcytotoxicity test. Here's a look at the equipment needed for PCR. This is the instrument that performs the PCR testing, and then the agarose gel in order to visualize the results. This flowchart does a nice job of summing up what goes on in terms of compatibility testing for donor and recipient for the purpose of transplantation. First and foremost, we need to make sure donor and recipient are ABO compatible, so we have to do ABO testing. We want to test each for the presence of cytomegalovirus. If the recipient is CMV negative, we wouldn't want to give them tissues from a donor who is CMV positive. We test both donor and recipient for their HLA antigens via the PCR test method. If all of this is indicating a go, then if everything's working out well, then lastly we do a cross match, which is recipient serum with donor cells to see whether the recipient has any preformed antibodies that are going to react with donor cells. In the case of a donor who's deceased and the family has donated the organs, then the results of the ABO, HLA, and CMV results go into the national database to try to find a recipient that would be compatible. This slide is showing us the results of PCR testing, whereby the DNA fragments have been amplified via PCR. Then the amplified fragments are separated by electrophoresis. So They'll separate out according to size, so each one of these is a different patient. Then the fragments are stained with ethidium bromide, which fluoresces under a UV light. The size of the DNA fragment can be estimated by comparing its migration pattern to fragments of DNA that are of known size. Because the sequence of the DNA fragment is known, its molecular size can be determined. If the size of a fragment amplified by PCR corresponds to the size of a fragment estimated by sequencing, that DNA fragment is confirmed to be the one amplified by the PCR primers. We've mentioned this before, but it bears repeating. Patients that need transplants may have antibodies to HLA antigens that would interfere with the success of the graft. So these HLA antibodies could have been stimulated to develop because of previous blood transfusions. Could be the patient has been pregnant and developed antibodies to fetal HLA antigens, or the person could have been previously been the recipient of a transplanted organ or tissue. So obviously, once they've formed particular anti-HLA antibodies, we cannot give them the corresponding HLA antigen again, or the graft will be a no-go. Indirect enzyme-labeled immunoassays, ELISA or ELISA, can be used to detect the anti-HLA antibodies. Basically, there's microwell plates provided by the kit manufacturer. The plates are coated with various HLA antigens. Then the patient's serum is added and allowed to incubate with the HLA preparations, and then we observe to see whether there's been a combination of antigen and antibody. If using multiplexing to determine HLA antibodies, a bead set of one color is coated with a set of HLA antigens derived from one individual of a known type. A bead set of another color is coated with HLA antigens of a different individual. Serum from the potential recipient is incubated with the beads. If the recipient has antibodies to a set of HLA antigens, these antibodies will bind to the bead to which those antigens are attached. After allowing the incubation step, we wash away to remove any unbound components. Then the beads are incubated with anti-human antibody, and that is fluorescently labeled, and then we can run that through a flow cytometer. And in the flow cytometer, we're detecting the fluorescence given off by the anti-human globulin, and we're evaluating the bead color to be able to determine which HLA antigens are involved.